My name is Andrei Herdean. I'm a postdoctoral researcher at University of Gothenburg, and I do research on the topic of photosynthesis and different aspects that are connected to this mechanism. This project was started in November 2016, and at this point in spring 2017, we're still in the beginning of the project, where we're still creating the mutants, and we're expecting that we're gonna be able to come to a conclusion of the project sometime during mid-2018. The idea is fairly simple. You have two genes nowadays that have two different functions, but they have a common ancestor. So somehow during the evolution, one of the genes acquired a new function that didn't exist in the past anymore. So we want to study this, and we want to show the mechanism by which this happened. We're going to recreate the gene that is extinct, that doesn't exist anymore, that disappeared about 40 million years ago. We're going to synthesize it, put it back into plants, and study how the function of that gene relative to the two new ones that we have nowadays. And by doing this, we can actually bring experimental proof to the mechanism that actually creates new functions in evolution. So it's going to be very exciting if it works out. The methods that I use in my research projects are non-invasive ways of observing and measuring how photosynthesis functions without actually disrupting the plant or the organism itself. And these methods are chlorophyll A fluorescence, electrochromic band shift, and P700 absorbance changes. By using this small number of tools, we learn a lot of things of how plants are doing while we study various aspects of their biology. All the research that I am doing currently is being carried out on the model plant Arabidopsis thaliana. The advantages of this is that we have many knockout mutants that we work with, and it's easy for us to engineer new genes into this plant. The plants are grown in the facilities here at the department, in growth chambers that where we either simulate conditions, at least light conditions from outside, from nature, where the light composition and fluctuations vary greatly, or we grow them in what we call standard conditions, under light that it's not damaging or hurtful to the plants. Yeah, so if this project actually works out and we can actually prove that neo-functionalization and positive gene selection is what created this gene that we're studying. If this project works out, this can become really a textbook example. So this can actually end up in the manuals for students that study evolution. But yeah, studying evolution, it's not a trivial thing, and it's very difficult to bring experimental evidence for mechanisms that take millions of years, and that's something that I'm trying to accomplish with this project. So show experimentally within 2 years what evolution has accomplished over the course of 40 or 60 million years with these two particular genes. And then the second very exciting project has to do with solving the network of proteins that exist in plants to let salts get in and out of the plants. And these proteins are ion transporters or channels. So these are proteins that sit in membranes and allow ions to go from one side of the membrane to the other one. So what I want to do is find as many as possible of these proteins that mediate these ion fluxes. And I'm mostly focused on transporters and channels that are located in the chloroplasts and affect photosynthesis. Again, the importance of understanding how these ion transporters and channels work in the plants, this has value for future crop engineering. So this can actually have direct applications in providing food in parts of the world where you cannot grow crops because of various pollutants. By finding these genes, we're gonna gain very important knowledge that could later on be used in crop engineering. So if you know how salts get into your plant, then you can engineer that particular plant to survive much better on soils that are contaminated with various salts.